So our next speaker is a research scientist in water desalination and reuse center. She has worked anaerobic granular sludge treatment in her PhD, and also she moved to EPF Lausanne and worked there on the aerobic granular sludge treatment for the domestic water. So she worked with the domestic water and industrial water both for aerobic and anaerobic wastewater treatment. And she's also one of the fellow of the prestigious Marie Curie scholarship. Here at CAST, she is involved in many projects. One of the projects she will talk about today about the microbial life under reverse osmosis. But a part of that, she's also involved in anti-scalants microbial growth potential under seawater conditions. Also, she's involved in estimate in situ microbial growth in drinking water taps. So please welcome Dr. Graciela Gonzalez-Gibbs.
Thank you, Mustaqim, for the introduction. Thank you, everyone, for being here. I know the weather is getting very nice, actually, and then probably you would rather be in the beach. I hope you enjoyed the talk. Under the sea, scientists have discovered underwater pools. These pools are made of very, very salty water. The salt content in these pools can be 8 times higher than the salt water in the upper seawater. Therefore, they are called brine pools. Brine pools are death traps for most organisms. For example, for crabs. Here, for example, if I will just analyze DNA sequences from samples taken from this location, and without doing a deep investigation, and here literally a deep investigation, I might wrongly conclude that crabs live in brine pools. Microorganisms have a better chance to survive in these hostile environments. They might live at the interface between the brine and the upper normal seawater. However, life under these hypersaline conditions is not easy. It's energetically expensive because microbial cells need to counteract the osmotic pressure of these environments to avoid plasmolysis. Hey, you might think you are in the wrong seminar. What all this has to do with the reverse osmosis? Well, in reverse osmosis, seawater is passed through a tube at high pressures, and this tube contains semi-permeable membranes. When the seawater is passing through, then we can recover fresh water. In many places in the world, this is the only way we can get fresh water. However, the process is not 100% efficient. And therefore, the remaining water, or the remaining seawater, is very, very salty. And this reminds us of the brine pool, right? Then it's not only that we have this gradient of normal seawater and very hypersaline conditions in the reverse osmosis system, but also just at the interface of the semi-permeable membrane, because there is a stagnant water layer there, we can have micro brine pools. And because of this analogy, I really start wondering, well, I asked myself this question: how do seawater microorganisms react to these salty conditions of these man-made brine pools? We know that the seawater that is entering into the reverse osmosis systems here is not sterile, despite the efforts of pre-treatments. So we want to know what will happen to these organisms if they enter the system. We don't want them to accumulate, and worse, we don't want them to grow on top of the membranes. My hypothesis was, or is, that the growth of seawater microorganisms diminishes as the water gets saltier. To test this hypothesis, then, of course, we need to do experimental work. And therefore, we went to the desalination plant here at KAOST, and we went to collect inlet seawater and outlet brine. We measured the conductivity of these solutions. Please see here, 60 millisiem over centimeter, and here, 90 millisiem per centimeter. Then we set up an array of salinity gradient that mimics the reverse osmosis system. So we can see here, for example, 60, conductivity 60, and here 90. Then we can make a mixture of seawater and brine and get 50 and 50% of each and get this conductivity. And then 2 more values in between. We then can set up an experimental array with a series of vials where we can investigate how microorganisms grow under these conditions. We set 3 biological replicates, and here, just look carefully, all of them have seawater. Then we set up this gradient of conductivity by adding sodium chloride. After that, I will not go into all the details on how we get up to get all these vials, but at the initial time, we get, for all of them, a similar concentration of microorganisms. Here I just said bacteria, but in fact, those who are microbiologists will say, oh, it's not only bacteria, of course, it's also archaea. Then we incubate at 30 degrees Celsius, and over time, we count the bacterial cells in these vials. What did we get? Well, here you can see the number of cells over time for the different treatments. Here is just the seawater with 60 millisiems over centimeter of conductivity. And then as we move along the conductivity gradient, here we have seawater with this conductivity similar to the brine at the desalination plant here at KAOST. We do see that the microbial growth decreases at high salinity. And of course, the data is the symbols, and this is a model, a logistic model for the microbial growth. If we extend the experiment, because I was thinking maybe when the microbes are exposed to this high salinity, they can adapt, and then they can still grow. But that was not the case. The growth starts to decrease, and then they start to die. And similarly for the other vials. Then I thought, okay, but those conditions were a bit artificial, because only water was added to get the conductivity of the brine. So what will happen if we do the same experiment, but doing it in a realistic or more realistic way by mixing the seawater actually with the brine of the plant? Here we set up again a series of vials to see how the microorganisms will grow under these conditions. We have here 100% seawater, 100% brine, 50 and 50, and 2 values in between. We have exactly the same conductivity range as the previous experiment, and we added at initial time the same or close cell content values. And we measure the cell content or the growth of bacteria over time. We got the same trend. And we can see the brine conditions negatively impact growth of seawater microbial cells. And the growth rate is about 3 times lower in brine than in seawater. From these results, we can calculate the growth rate, and we can see that as the conductivity increases, the growth rate decreases. And there's a linear high correlation between these 2 parameters. So this doubling time, this growth rate represents about 5 hours of doubling time for microbes growing just Red Sea microbes. And when these microbes are exposed to brine conditions, then the doubling time increases 3 times. how do these data compare to literature data? At the beginning, when I had this question, I started to look in the internet, and I did not find any information about how microorganisms will be impacted at the salinities that we find in reverse osmosis system. And this is why I started to think about conducting these experiments. Here we have the growth rate over salt content of the Red Sea bacteria. That's this experiment here. I compared this data with these three microorganisms. These three microorganisms are allophilic microorganisms. That means microorganisms that like to grow under very salty conditions. This one is a bacterium, and these two are archaea. So we do see that even these archaea that love to live under very salty conditions, their growth rate is very much low when the salt concentration is quite high. Good. Now, we might think, okay, or I was thinking, maybe this is very obvious. It's so obvious, but it seems that not everybody or not many people were thinking about this in practice in reverse osmosis systems. And of course, then one can say, oh yes, it's obvious, but it's obvious when somebody points it at you, right? Now, the question for me and the most important thing now is, what do we do with these findings? What do we do with our learnings? What do we learn? Well, we learned that microorganisms from the inlet desalination plant grow relatively fast. Their doubling time is about 5 to 6 hours. And we learned that increased salinity, as the salinity found in a reverse osmosis system, inhibits microbial growth. So the growth rates are lower, 3 times lower compared to the inlet seawater. Now we need to exploit this knowledge. And one of the important things is that we should expect low microbial growth at high salinity, particularly at the membrane interface, or the interface between the membrane and the stagnant layer within the reverse osmosis system, because in this location, the salinity content can be even higher than the brine salinity content. And I think this may challenge previous thoughts. In fact, in one report I read, people suggesting that specifically in this interface, you might have higher growth because you have concentrated nutrients. But you have to think that you have a very, very concentrated salt seawater, seawater with salt. Then, because seawater microorganisms grow very, very fast, then we have to ensure that all storage tanks should have a much lower residence time compared to the microbial doubling time. And this is particularly important in those desalination plants that use ultrafiltration membranes. So they can do ultrafiltration, but nevertheless, the efficiency is not 100%. So if the membrane still can pass through some microorganisms, and then they store the water for maybe 6 hours or higher times, then they will end up having just a culture of microbial cells in the tank. And it will be detrimental for the reverse osmosis membranes. Then the next thing we can discuss is relates to the new idea, to a newer idea that is called follow reversal to minimize scaling. So people propose that if you have the reverse osmosis membrane, as soon as you detect that the salinity concentration reaches a certain level at the outlet, then to avoid precipitation of salts, they reverse the flow. So then you have here a lower concentrated seawater. Then you avoid precipitation. Possibly, one will have to think and maybe do some experiments. Maybe it's possible to also, at the same time minimize microbial growth. but this needs investigation. This is just a thought. Then the other idea is whether we can use osmotic shocks to inhibit and then clean microbes on the membrane. And when I was thinking of this, yes, I think it might be possible. And then I was thinking, well, if we just inhibit the microbes, maybe they are not dead. Maybe we don't kill them. We don't destroy them. We don't break their cells. So then will it be easier to clean a dead microbe or a live microbe? This question might look weird and maybe nonsensical, but maybe it's important to think about it. And well, sometimes key information is more hidden. And in order to find this information and to make desalination more efficient, we really need to look more carefully and deeper. And with this, I would like to thank you for your attention. And in fact, this talk the purpose was to show you our most recent findings. These findings just literally came out of the oven a few weeks ago. And it will be nice if we have just like a tsunami of thoughts, and then we can discuss some possibilities. And yeah, I will be happy to answer any question you may have. And thank you for your attention.
Thank you, Dr. Graciela. Thank you very much. Very inspiring talk. And really, the presentation is really amazing. I will ask your help whenever I start my presentation. Thank you. So the floor is open for the questions. So for the people who are physically joined, so please raise your hands, and then you can ask the question directly. Yes, please, Theo.
Yes, the microbial communities we have in the samples are the microbial communities that you will find that they will enter into the desalination plant. Now, if I would like to know the taxonomic names of the microorganisms, then we will do these experiments afterward. This is another step. Now, the interesting thing in asking which microorganisms do we have, I see it in relation to knowing whether we have, what is the proportion of allophilic microorganisms that we have in the seawater. So maybe the number of allophilic microorganisms in the seawater, from when you take it out from the surface, as is taken to go to the desalination plant, maybe it's not so high as if you would take a sea sample below, I don't know, 500 meters or 1,000 meters below the seawater, close to a volcano or these hydrothermal vents. That will be different. But in any case, I think even if we will have allophilic and mostly allophilic bacteria are also thermophilic. So even if we will have some, it will be hard for them to grow within the arrow system, I think, because the arrow system is not thermophilic. I mean, it's not super thermophilic. And also, if you would like to think, okay, maybe you can enrich them, we will have to think about the retention time of the water in the arrow membrane, and also if they attach, what will be the retention time before people apply cleaning processes of the membrane. So it's a complex and it's an interesting system to study, I think. And I think it also, although some people might think, oh, it's very much already optimized, I think there are still some points that we can optimize further if we apply, in this case, microbiological knowledge to further optimize the system.
Okay, so any other question physically? Yes, please. Yes. Yeah, Victor, please introduce yourself because there are a lot of people.
Yes. Yes, definitely. And thank you for the question. This is a very, very key point, because I think exactly, there is one of the points where we can exploit applying the knowledge to improve the system, to make it more efficient. So definitely there are anti-scalants that people apply just before entering the RO system. We are doing some studies to investigate how much growth can you get out of these anti-scalants. And there are different types of anti-scalants. They are different chemically, and they are different in the response to promote bacterial growth. Maybe in another talk, we can talk about that. But also, I think in this, knowing that high salinity contents inhibits microbial growth, then it just came to my mind yesterday. So if we have these long tubes of the RO membranes, mostly salt precipitates will occur when the seawater is more concentrated, so close to the outlet. But nevertheless, people add anti-scalants at the beginning. So will there be a way to design a system where you add anti-scalants not at the beginning, but somewhere in the middle? I don't know, maybe it's a crazy thought, but why do we want them to add them where they are not needed? Maybe it's possible to add them afterwards. And even there's another question. Do we really need anti-scalants? I think I have tried to look into literature for some of my questions, and I just cannot find the answer. And one of the questions is this one. Mm-hmm. Mm-hmm.
Okay, so we have one question from our virtual audience from Zoom. So please unmute yourself and ask the question directly.
Hello, this is Dr. Ehsan. Actually, my question is regarding these microorganisms or traces. Is there any possibility that it may end up to the concentrate or brine? This is the first question. And if yes, whether it can make the brine more hazardous or no?
I didn't get the answer.
So, Dr. Hassan, could you please ask, I think you are asking the second part of your question, that those microbial microbes, do they make the brine more hazardous or not, right?
Yes, correct.
Okay. So the microbes, do they make the brine more hazardous? This is the question, the second part.
Yes. Well, under these conditions, we would not expect that pathogenic microorganisms will grow on the brine. First of all, they will have to come in the inlets, and the inlet is mainly seawater. So pathogenic microorganisms, the seawater is not their natural environment, unless, of course, you can find pathogenic organisms in beaches, in the beach, but this is because they arrive to this location because of wastewater discharges, or yeah, mainly. And the inlet from the desalination plants is located far away from beaches, so we don't expect these organisms to be there.
Okay, thank you. So I hope, Dr. Hassan, you get your answer.
Okay, thank you.
Okay, so do we have Claudia, another participant? Okay, from Zoom. Okay, so I have one question, quick question, Dr. Graciela. So we work in the water desalination in thermal, for example. So you discuss about the reverse osmosis. So which system is the best system to desalinate water if we have such system, sub problem of the microbes?
Well, if you ask about the microbes, and then we just learned that if you have over the system a very salty solution and this inhibits microbes, then that will be I would think, a good point. In other systems, I don't know if you reach this level of salinity that would inhibit microbes. You can say, okay, maybe in other systems like thermal desalination, you do have conditions of high temperature. And at first glance, one might think, okay, at high temperature, you might inhibit microorganisms. Yes, you might inhibit mesophilic microorganisms, but definitely you will find, and it's very likely that you have the growth of thermophilic microorganisms.
Okay. Yeah, thank you very much. And if we don't have any question, I think, is there any question from Bex? Okay, if not, so we would like to thank Dr. Graciela and Dr. Saqib, and also we would like to thank all the support from the center, and yeah, all of you people from whether you are joined through virtually or physically. So thank you very much, and please stay tuned with us on every Sunday from 3 to 4. So next Sunday we have very interesting presentation from Professor Bruce Logan, who is traveling from US, and everyone knows who is Bruce Logan. So please stay tuned from to next Sunday from 3 to 4 p.m. And so we have also outside the center is so happy with us. So they put some of the refreshment in the brown bag. So please feel free to pick the brown bags. So we have enough brown bags for everyone. So yes, the last, please thank you to all of both of our participants, and yeah, thank you both of them. Okay, yeah, so there is another announcement. We have a internal water talk from the IWA tomorrow. So it's the same room. So this will be more on the sustainability of the water. And the Vice President of Cost, Facilities and Management will come to discuss about the water treatment and wastewater and some other aspect of the water. So please feel free to join us. It will be recorded, and it will be here from 5 to 6.